today's webinar, Using In Silico Toxicity Models for Industrial Research and Regulatory Evaluation. Michael Lawless, Team Leader, Cheminformatics Studies, will be presenting today. Joining Michael and myself is Anu Sharma, Senior Marketing and Sales Manager, and Dr. Bob Clark, Director of Life Sciences. An opportunity to ask questions will follow Michael's presentation today. You may either send your questions using the questions pane on your control panel, or you may ask your question directly using the hand-raising icon. If you're using a telephone to listen to the call, please be sure to enter the unique audio pin displayed when you join the call. When you enter your audio pin, this enables us to unmute your line so that you can be heard. This webinar is being recorded for playback at our website, www.simulations-plus.com. It is now my pleasure to turn the presentation over to Anu, who will provide a brief introduction to our company. Anu?
Thank you, John. My name is Anu, and I'm the Senior Manager for Sales and Marketing. I would like to first introduce you to our company and the product. We were founded in 1998. We are located in Lancaster, California, which is about 1.5 hour drives from Los Angeles. Ours is a publicly traded company trading at NASDAQ with the ticker symbol of SLP. We offer software for simulations and modeling, an estimation of physical, chemical, and biopharmaceutical properties and ADMET properties. Our software is used for analysis of QSAM and for prediction of in vivo absorption, pharmacokinetics, and pharmacodynamics properties. On the site, we also offer services for the small and big projects. And as you see on the right-hand side, these are our products: Gastric Plus, ADMET Predictor, Metchem Studio, Ddv Plus, and Metchem Designer. In short, we will help you make decisions faster. Simulation Plus offers value at every stage of the drug discovery processes. As you see on this slide, we offer Metchem Studio and Metchem Designer for the medicinal chemists in the earlier stages of the drug discovery program, and for the biologists, we have Gastric Plus, and for formulation scientists, we have Ddv Plus. ADMET Predictor comes as a standalone product as well as a module which could be integrated for the medicinal chemists in the Metchem Studio, Metchem Designer, and for the biologists in the Gastric Plus as well. I will go into the brief details on each of our products now. Gastric Plus is our flagship product, and it was founded in 1997, and since then we are offering the industry's most accurate, flexible, and powerful simulations program. Gastric Plus allows you to expand the computer simulations into the realm of absorption, pharmacokinetics, and pharmacodynamics. It allows to understand the behavior of drug candidates in animals and humans. ADMET Predictor, as the name sounds, it is a product that stands for the acronym at absorption, distribution, metabolism, elimination, and toxicity. ADMET Predictor allows to estimate a number of vital ADMET properties from molecular structures. It allows for building predictive models of new properties from users' data. ADMET Predictor comes in a package of various modules: physical, chemical, and biopharmaceutical properties such as pKa, log P, log T, polar surface area, solubility, etc. It also comes with a metabolism module, which will allow you to predict the likely sites of metabolic attack of human cytochrome P450 We also have a toxicity module in the ADMET Predictor, and Mike is going to be going in details with this product. Metchem Designer is our free tool which is used by the scientists to design the structures, and also in this free tool you will be able to predict all of the Lipinski values. But with the paid license for ADMET Predictor, you'll be able to predict more than 140 properties and the likely set metabolites. MedChem Studio is a product for the medicinal chemists. It is used for data mining and drug designing, and it offers support in lead identification, de novo designing, scaffold hopping, and lead optimization. It has features for R group decomposition and for SAR analysis, and it allows for building queries and designing drug capabilities based on reaction processing. Last but not the least, our first and the only simulation tool for in vivo resolution experiments is the Ddv Plus. It's the software tool that models and simulates the in vitro resolution of APIs and formulation experiments dosed as powders, tablets, capsules, etc. under various experimental conditions. With this, I would like to introduce you to several of our valued customers who range from the non-profit organizations, academia, small and big biotech companies, pharmaceutical companies, and various other industries as well. And with that, we would like to welcome you aboard. I would like to introduce the next set of slides to Michael Lawless, who will be going into the details with the toxicology module of ADMET Predictor. Over to you, Mike.
Thank you, Anu. Okay, so this next slide is simply an outline of the talk. So first I'll give an introduction to toxicology, discuss modes of action, chemical classes, and toxic anatomy. Next, I'll describe creating artificial neural network ensemble models and show an example of building a developmental toxicity model. This will be followed by a detailed discussion of the models in our ADMET Predictor toxicity module and show an example of predicting human liver adverse effects. And then in the final section, I'll discuss tox risk, which is a liability score for potential toxicities of compounds, and show an example of creating an environmental risk score. The goal of in silico toxicity modeling is to accurately predict toxicological endpoints. This will help to anticipate potential toxicity problems before synthesis, and also reduce and replace animal testing. It was Paracelsus in the 16th century that made the correlation between dose and activity, in his statements that all substances are poison, there is none that is not a poison. The right dose differentiates between the poison and the remedy. Even a compound as simple as water can be toxic if we drink enough in a short amount of time. Another example is vinyl chloride, which at very high doses is an hepatotoxin. At lower doses over a very long period of time is potent carcinogen, and then at extremely low doses is harmless. So some of the modes of actions or areas of study in toxicology are shown on this slide. So carcinogenicity deals with the events leading up to cancer. Teratogenicity relates to birth defects and developmental defects throughout the lifetime of the human. Mutagenesis deals with events leading up to and damaging the genetic material in organs or organisms. Organ toxicity is specific to organs such as liver, kidney, heart, etc. And then biochemical and molecular toxicology deal with the interactions of toxicants with proteins and DNA, and gets down on the molecular level. This next slide just discusses the different classes of chemicals that are potential toxicants. So first on the list is agricultural chemicals. This includes things like pesticides, insecticides, herbicides, etc. Pharmaceuticals, both especially in side effects of pharmaceuticals. Drugs of abuse such as cocaine and heroin. Something that's kind of new recently is this K2 synthetic marijuana, which has sent several people to the hospital. food additives, metals, solvents. Of course, cosmetics would be an area of interest. Obviously, you wouldn't want those causing any type of skin allergies, etc. Industrial chemistry, chemicals, naturally occurring substances, and then combustion products include things from the petrochemical industry. This slide describes the fate and effect of toxicants on the body, starting off with exposure. Exposure can be intentional, or it can be associated with your occupation, or it can be exposure from the environment. Different ways that the organism can be exposed to toxicants are ingestion, which can be deliberate or unintentional, inhalation, which would be maybe occur in an occupational area, and then compounds can also permeate into the skin and get into the system that way. So once the toxicant is absorbed or exposed, then it will be absorbed and distributed throughout the body into various tissues and organs. If it goes into, for example, the liver, it will be metabolized and then sent back into the circulating blood system. Eventually, the toxicant is going to interact with DNA or proteins in the cells and cause the toxic effect. And eventually, it will be excreted. So this is the overall fate of a toxicant in the body. And all these areas are important to toxicology. Okay, there are a number of regulatory industries, agencies that are using in silico predictions to support their regulatory decisions. So, for example, the United States Environmental Protection Association, the Food and Drug Administration, and here was a nice article published this year on their QSAR models and how they assess the safety using in silico techniques. The FDA also uses in silico predictions to avoid additional review when there is data on a chemically similar compound. So in their database, they have a large number of chemicals, and if a new chemical wants to go through the regulatory process, and it's similar to one that's already there, you can use information from that, and your models are going to be more accurate, of course, if you have similar compounds in your data set. If some of the safety information is unavailable, you may predict the outcome of that experiment. You can also identify compounds that need further toxicity testing, or when a safety study is equivocal. This next slide just kind of outlines building artificial neural network models. The starting point is structures and experimental data. It's important that the structures are in the right tautomeric form. Experimental data has been validated and verified. If you have two different experimental results for the same compound and they differ greatly, then you need to resolve which one's the more applicable to your model. The next step is to compute descriptors for the compounds in your data set. We include over 300 descriptors in ADMET Predictor, and they're available for model building. I just want to emphasize the charge and reactivity descriptors. So we have an EEM Huckel charge model that was parameterized from ab initio calculations. So we can very accurately and very quickly predict charges that are very close to ab initio results. This allows us to also compute Fukui indices to deal with reactivity. The next step in the process is to remove low and correlated descriptors from the descriptor pool. So for example, if a descriptor doesn't vary from molecule to molecule, it's not going to be able to differentiate the biological activity of the molecule. So we would simply drop that descriptor. We also perform pairwise correlations of the descriptors, and if two descriptors are highly correlated, then we would drop one of those descriptors because we don't want to introduce redundancy into the descriptor space. The next task is to divide the test set or the data set into a test set and a training set. We have various methods for specifying the test set. And so once we define the test set, it's put aside and sequestered. So this is truly an external test set, so because we don't train on the test set and we don't cross-validate. The remaining molecules go into a training pool or a training set, and at this stage we need to specify to the program how many neurons and descriptors we're going to use. Now, a priori, we don't know the number of neurons and descriptors that will create the best models, so what we do is experiment and we create a large number of models with different architectures or different numbers of neurons and descriptors. We also need to, since we're reducing the number of descriptors, we need a way of selecting what descriptors are going to create the best models. So this is called sensitivity analysis, and we have a number of different methods to select descriptors. Then we train the models based on the training set and building artificial neural network ensembles. Once the models are built, they're applied to the test set, and then we can compare the statistics of the test set and the training set in order to pick the best model. Our models are ensembles of 33 artificial neural networks. Two types of models are used in our toxicity module. One is a classification model and the other is a regression model. Shown on the left are the results of a classification model. So we have a number of compounds with observed activity, in this case non-toxic. We're classifying them as either non-toxic or toxic, and then creating predictions via the model on the x-axis. So compounds down in the lower left-hand corner are true negatives. So these are compounds we predicted to be negatives, and in fact they are observed negatives. Over in the upper right-hand corner, these are compounds that we predicted to be positive, or predicted to be toxic in this case, and in fact they are experimentally toxic. So then in these two corners, these are the compounds that we incorrectly classified. So here we predicted these compounds to be positive, but they're actually non-toxic. So these are our false positives, and up here are our false negatives. And there are a number of statistics we calculate for the model in order to compare different models to each other. So one is specificity, the other sensitivity. The Gowden index is a balance between the specificity and the sensitivity, and then the false rate is simply how many of the predictions are incorrect. So with specificity, we're looking at the number of correctly predicted negatives divided by all the observed negative values. Sensitivity looks at the number of true negatives in relationship, or in the ratio to all of the observed positives. And these two are important because if you have an unbalanced data set, say that has 90% non-toxic and 10% toxic, you could simply say I'm going to predict everything to be non-toxic, and you'd be correct 90% of the time, but you'd always incorrectly predict any positive compound, and so your sensitivity would be 0. So it's important to look at these other statistics. Over on the right-hand side, we show an example of a regression model. So here we're trying to accurately predict the log P. Here are predictions on the x-axis, and as you can see, these fall on a line with slope close to 1. And we have a very large test set in this case of over 12,000 compounds. The root mean square error is about 0.3 for both the training and the test sets, and we have a correlation coefficient of 0.97. So some other statistics are listed down here that we use to judge the model. Okay, another important aspect of in silico predictions are defining the applicability domain. So you want to be able to predict compounds that are similar to your training set, but if something's very different from compounds in your training set, you're not going to be able to, in many cases, produce an accurate prediction. So the way we define our applicability domain is we look at the training pool of compounds and the descriptors space. So in this example, this would be just a simple 2-descriptor model. The compounds in the training set would have maximum and minimum values, of course, spanning the space. And then we add a 10% buffer all the way around this, and that's what we define as our applicability domain. So when we make external predictions, as indicated by these gray points, all these fall inside the applicability domain, so these compounds are in the scope of the model. Now, if we try and predict compounds that are outside the scope, so for example, this one out here, it's above the maximum value of anything in the training set for descriptor 2, then we would classify these as out-of-scope predictions, and we would color the value in ADMET Predictor in magenta. Okay, so I just want to demonstrate creating an artificial neural network ensemble model of developmental toxicity. So developmental toxicity refers to any abnormality before or after birth of the developing species. So this could include the death of the fetus, growth retardation, reduced body weight, organ toxicity, or some kind of structural defect, some kind of birth defect. Of course, this is a very challenging model because of all these multiple mechanisms involved in the toxicity. So what I'm going to demonstrate is just taking a subset of the compounds that we use to build our reproductive developmental toxicity model and show how we would build a model. And this is the reference for much of the data that we used to create the model. Okay, so this is ADMET Predictor, and I've simply read in 120 out of the almost 300 molecules for the developmental toxicity model. And so this is the read in the structures, and then I have a column that indicates whether the compound is a developmental toxicant or not. So the first thing to do is to calculate the descriptors. So I just use this pull-down menu and click Calculate, and so very rapidly it calculates over 300 descriptors for these 120 molecules, and it also calculates over 100 models in ADMET Predictor. So the first thing I like to do is get some kind of idea of the distribution of the data. So I go into our histograms tab, and you can see that about 60% of the compounds are non-developmental toxicants and 40% are developmental toxicants. So this is a fairly balanced data set, 60-40. The next step is to go into the basic modeler set tabs and specify the dependent variable, which is developmental toxicity in this point. So over in this right-hand side, we have 327 descriptors available for training. If we want to add any of these descriptors over on the left, we would simply highlight them and then press this button here, which would transfer them over to here. Similarly, if you had your own descriptors that you wanted to use to build a model, you could read them in with the initial file, and then they'd be available for modeling. The next step is to go into the Advanced Modeler Settings tab, and then we have various sub-tabs down here along the bottom of the screen. So I'm going to specify a minimum test set size of 10%, punch in my favorite random number, and I'm going to use a Cahonen self-organizing map to define the test set. The next step is to define which method we want to use for our sensitivity analysis, and I'm going to use a genetic algorithm and ask it to apply the genetic algorithm each time it creates a new architecture. Then the final step is to specify the number of neurons and descriptors that I want to create different architectures for. So I'm going to start with 1 neuron, go up to 5 neurons with a step of 2. So it's going to create 1, 3, and 5 neuron models. I'm going to specify a minimum number of 20 descriptors and a maximum, or minimum of 10 and a maximum of 20, and then finally a step size of 3 on the number of descriptors. So then going back into the basic modeling tab, I simply click on the ADMET button. The first thing it does is prompt me for what I want to define as the negative set, and I'll say the molecules that are classified as no go into the negative set. So it starts building models, as you can see, and I'm just going to view the log file very quickly. So for every time we create a model, we have a lot of files that are produced. These go into a particular directory, and one of them is the log file. So the first thing it does is look at descriptors that have low variance, and we'll just throw all those out. Similarly, things that are underrepresentative are also thrown out. Then the next thing the program does is compare each descriptor to every other descriptor, does a pairwise comparison, and if they're highly correlated, then we drop one of the two descriptors. So in this case, we end up with 181 candidate inputs, and then the next thing we do is go and create this Kahona map. So with the Kahona map, it basically clusters compounds together based on the descriptor space and puts them into cells. The first compound in the cell would go into the training set, the second would go into a verification set, and then the third and any additional compounds in a cell would go into the test set. Okay, so if we go back to our model building process, it's finished building, and it built a 3 by 5 matrix. We can click Find Best Ensemble, and this uses some statistics based on the test of the results in the Alden indice and the training set to try and select a balanced model. And here it's selected a 3-neuron, 20-input model. We can look at the sensitivity, which is very good for this particular model, so 0.91 for the training set and 1 for the test set. Specificity, a little bit worse, 0.73 and 0.82 for the test set, the Alden index, and then finally the false rate. If we click on a cell, we can see this confusion matrix that I showed early in the slide. We can click on a point and see what the compound is and then what the prediction. So here predicted no, and it was actually a positive test. So that's a compound we incorrectly predicted. I can also, if I decide that this is a nice model, I can export that model and then save it and make it available in future versions or future uses to perform predictions. Okay, so before I leave this, I just want to show the grid for the bigger data set. So we actually had 292 molecules in the data set, and we produced a very large grid of models and then selected the best model there. Okay, so now going back to the slides, in this next section I want to describe the models in our toxicity module of ADMET Predictor. So models can be classified under these 7 categories, and then we have several models under each category. So starting over on the left, for cardiac toxicity, we compute the if it will block the hERG channel. For hepatotoxicity, we look at 5 liver enzymes and if they're elevated in serum. Endocrine system, we look at estrogen and androgen receptors. For carcinogenicity, we have models related to rat and mouse TD50s, immunogenicity, and chromosomal aberrations. For sensitization, we look at skin and respiratory sensitization, and then environmental toxicity, we look at bioconcentration factor, and then lethal dose for fathead minnow, water flea, and tetrahydropyroformis. And then 4 additional models which really don't fit well into these other categories are acute rat toxicity, developmental toxicity, which I just discussed, maximum recommended therapeutic dose, and then finally phospholipidosis. So in these next few slides, I'll describe each of the models in a little bit more detail. Overall, we have 34 models in our toxicity module. For the endocrine system, as I mentioned, we have estrogen and androgen receptor toxicity. First of all, we have 2 models that are classification models for both the estrogen and the androgen receptor. And so these are going to result in the prediction of non-toxic or toxic. If you end up predicting the compound to be toxic, then we have a quantitative model which predicts the relative binding affinity, which is a ratio of the IC50 of the reference ligand used for either one of the receptors relative to the IC50 of the test compound. So if it's a very potent compound, the IC50 of the test compound is going to be lower than the reference compound. So the higher this value, the more potent we bind to the receptor. This next model is based on the maximum recommended therapeutic dose. This came from a paper from Ed Matthews at the FDA. And Ed looked at about 1,800 drug molecules, and from the label on that extracted what the maximum therapeutic dose. And he noticed a correlation that compounds that had a low recommended therapeutic dose tend to have more toxicity problems. So he created models on that. We read the paper and obtained his data set and curated his data set, and there were about 10% of the compounds had a little bit of a problem, so we resolved those problems and sent those structures back to Ed, and ended up building a model where the cutoff was 3.16 mg per kg per day. So if it predicts that the therapeutic dose is less than 3.16 mg per kg per day, this indicates that there's an increased potential for side effects. For carcinogenicity, we have Ddv for rat and mouse. So this is the dose where 50% of the animals develop tumors. We have classification models for Ames mutagenicity. We look at 5 different strains of Salmonella, and each one of those strains is with or without rat liver microsome activation for a total of 10 models. And then the prefix for each of these model names is toxmut, and then whatever strain it is at the end. We have a chromosome aberration model, toxcarb, cardiac toxicology model, which I'll describe a little bit more on this next slide, in a little bit more detail on this next slide. So the hERG channel is a human ether-agogo related gene. This encodes a potassium channel. If the drug blocks the potassium channel, it can lead to fatal cardiac arrhythmias. And so there are some drug molecules that have been pulled from the market because they inhibited the hERG channel. So testing for hERG blockage is now mandatory in the pharmaceutical industry. So the first model we have is a classification model. There were 839 compounds in the training set and 185 in the test set. And for each one of our models in our user manual, we'll show the statistics, a similar table to this one for each one of the models. The concordance, sensitivity, and specificity were very high for the training set and similarly for the test set. So this is a nice predictive model. And similarly, we created a quantitative model based on the PIC50 for inhibiting the hERG channel. So a little bit smaller data set because some of the compounds in the literature either had a value of blocking the channel or not. They didn't quantify it with a IC50. And so we get a R-square of 0.7 for the training set and 0.6 for the test set. So the way we use these models is the classification model is used as a filter. So if it's predicted to be non-toxic, you can basically stop there. If the compound is predicted toxic, then you would use the regression model to predict the PIC50 of the compound. And just want to note that when we built the regression model, we used all the compounds in the training set, not just the toxic compounds. Okay, so moving on and describing some of the additional models in our toxicity module. First is hepatotoxicity, and I'll discuss this in a little bit more detail at the end of this section, but when you go to get a blood test from the doctor, they'll look at elevation of 5 different liver enzymes, and that will determine if you've got any liver injury. Acute toxicity in rats is an LD50 is another model. Skin sensitization and respiratory sensitization are two separate classification models. And then finally, phospholipidosis. So phospholipidosis is a lysosomal storage disorder, and it's characterized by accumulations of phospholipids in the tissues of the body. So lysosomes are these organelles in cells, and they contain enzymes which remove waste material from the cell. So in individuals that have this lysosome disorder, molecules that are normally metabolized become trapped in the cells. Okay. The reproductive toxicity and developmental, reproductive and developmental toxicity module is next, our model and that I've discussed previously. And then we have several models for environmental toxicology, bioconcentration factor, which is the ratio of the concentration of the organism to the concentration of the environment. So this is, for example, useful for fish. Fathead minnow toxicity to the T. piriformis, and then finally a model of Daphnia magna. Okay, so let me just pop into ADMET Predictor again and show how we predict how these predictions are visualized in ADMET Predictor. So we have a tab down here that's the toxicity tab. So these are all the models in our toxicity module. Here's the maximum tolerated dose, or maximum recommended therapeutic dose, and the prediction is less than 3.6 or more than 3.16 mg per kg per day. You see these are the estrogen receptor models. So for this particular compound, it's predicted as toxic, but it's shown in pink. So this is an example of something that's outside the scope of the training set. As you see, if I hover my mouse over a particular cell, it comes up with a tooltip that gives a little description about what the model is, and then the accuracy for the class for the model. If it's a quantitative model, it will give the root mean square error for the model. So here we see respiratory and skin sensitization, fathead minnow, Daphnia magna, bioconcentration factor, the herd channel, rat toxicity, carcinogenicity in rats and mice, chromosome aberrations, phospholipidosis, and then these five here, tox alkyl, and these associated ones are associated with the liver injury model. And then we have the 10 models for mutagenicity, Ames, results of Ames test. And then finally over here we have the tox risk score, which I'll talk about in the final section. Okay, so discussing the human liver adverse effects in a little bit more detail. So the U.S. Food and Drug Administration Center for Drug Evaluation and Research has been collecting reports on human liver adverse effects since 1968. And the results are stored in two databases, the spontaneous reporting system and the adverse events reporting system. Of course, modeling these adverse effects is challenging because the number of reports from individual drugs varies. It also depends upon the length of the time the drug was marketed and how many patients took the drug. Also, patients may be taking multiple drugs, so instead of the effect being due to a single drug, it might be due to a combination of drugs. So we tried to kind of standardize the results in the reporting index. So this is the number of reports for a particular molecule divided by the number of shipping units. So this is essentially how many prescriptions were taken. The RIs fall into three classes. Inactive, so if it has an RI below 3, marginally active, it's between 3 and 4, and then finally active is greater than 4. And what we did was just create a binary classification model to classify it either as inactive, and then group these two together and call those actives. So we have models for each of the 5 liver enzymes that would be examined when you go and have a liver test. So, or excuse me, a blood test. On this next slide, I show the results of training of the models. So these are fairly small data sets of about 100 compounds in the training set and in the teams for the test set, but we do get very good statistics out of these models. So there is a book called the Handbook of the Drug Metabolism Handbook, and in there's a table that lists 26 compounds that are hepatotoxic. So I input that into our program and predicted the hepatotoxicity, how much the liver injury models for these 26 molecules. Now, 12 of the molecules were in our training set, and out of the 12, we correctly predicted that 10 of those, or 10 would cause hepatotoxicity. So we got 2 wrong. Of the remaining 14, we predicted hepatotoxicity for 7 of them. And then the remaining 7 are shown in MedChem Studio here, and the top one is sudoxycam. So while we didn't predict this compound to be toxic, we can take this into our MedChem Studio program, and we can compute the potential metabolites of this compound simply by selecting the M button here. So you see these various steps can produce these 2 compounds. And then we can click on the AP button to calculate all the ADMET properties for these molecules. And they're displayed in the table here. And as you see that we do predict that one of the metabolites of this compound can cause hepatotoxicity. And so this is consistent with the experimental, with the adverse reporting system. Okay, so now we go back into the PowerPoint slides, and in this final section I'll describe the tox risk. So we have a scoring system called ADMET risk that encompasses 3 components of risk. So the first risk is absorption, so this is related to absorption and pharmacokinetic properties. CYP risk, which includes which measures if a compound has high CYP clearance or causes CYP inhibition, and then finally tox risk, which I'll discuss in detail. So tox risk is a toxicity liability score, and it is a flexible scoring system and encompasses components of toxicity. We have 7 rules by default, and the way we create a rule is we calculate the properties with ADMET Predictor, and we use a subset of the World Drug Index. And so we calculate the descriptor values for the compounds in the subset of about 2,300 compounds, and then we create a distribution plot of that, and we set a threshold such that 90% of the drugs would pass this threshold. So if you're in the 10% over that, you would get a score, single point, for that adverse effect. So it allows someone to rapidly see potential toxicities via a single number, and it's a way to assess large numbers of molecules for potential risks. So here's an example of setting a threshold. So one of the rules in the toxicity module is it's predicted HERG IC50. So this is a distribution of values for the 2,300 compounds, and if you get a tox HERG score of above 6, then you're in the 10% of highest values for the 2,300 compounds in the drug. So we defined a rule, and we have a code of HE for HERG liability, and so if a molecule has a tox HERG prediction of greater than 6, it would be flagged by this rule. Okay. Overall, we have 7 rules. The first one is the hERG inhibition if the predictive value is above 6. Rat acute toxicity, rat and mouse carcinogenicity, liver adverse effects, both for elevation of glutamyl transferase enzymes and also a hepatotoxicity score. And then finally an Ames mutagenicity test, and so this is the score of 2. So if it's greater, if it has 3 or more positives in our set of 10 Ames models, we would flag the compound. So the HG and the the HP and the SG reflect the actual blood test, how their blood test results are interpreted. So hepatotoxicity is indicated if any one of these 3 enzymes are elevated, along with SGOT and SGPT. If both of these are elevated, then it gets to a risk of SG. And tox risk is greater than 2 for about 10% of the World Drug Index. Okay, so let me just hop again into ADMET Predictor and just quickly show how we can create a customized tool or customized environmental risk score. So I'll bring up the model editor, click on create an ADMET risk, and so I'm going to use 2 simple rules. The first rule is if it's an endocrine receptor positive, so I'll use the code EN, and then simply go down to the estrogen receptor filter and say that if that equals toxic, then the molecule would get a point. So I'll add that to the rule, and then similarly, if we predict that the androgen receptor filter, we need an or in here, so specify if either of these filters is positive, then the outcome would be a score of 1. So the androgen receptor, similarly type in a toxic value for that, add that to the rule, and then another rule is if fathead minnow score, that's toxic to fathead minnows, so I'll go down and specify the fathead minnow result, and if this is lower than 0.02, in other words, a low value, the compound is toxic at a low dose, then this would be flagged. So I simply save the scores as a file, or these rules as a file, and then exit from this window. So then it asks me how I want to define these, so I'll define an environmental risk model, and then also the code for these. And click OK. So it adds this to the model descriptions, and then I can go back in and recalculate the ADMET properties for these molecules, and then go into the user model tabs, and here I have the toxicity risks for these compounds, or excuse me, the environmental risks that I just created. I just want to briefly show some of the results from the tox risk on the set of about 100 compounds, and these are mainly pesticides and insecticides, so agricultural type compounds. So here's a compound that has a tox risk of 5. It is predicted to be carcinogenic to rats, mouse, cause hepatotoxicity, and also raising the SG and SOT and SGPT enzymes, and then also this MU here is for mutagenic. So this is a fairly nasty compound by our predictions, and I just want to stress that if our models sometimes can be wrong, but if several models are indicating that the compound's toxic, that's even more evidence of toxicity. So on the opposite end, if you get compounds that have no tox risk, or maybe a tox risk of 1, maybe we didn't quite do a correct prediction on this molecule, and so maybe this is okay. Maybe it might indicate that you need to do additional testing on that also. So then finally, just going back to the slides and summarizing with this final slide, the goal of in silico toxicity models is to predict endpoints of chemicals that can help to reduce or replace animal testing and also allow you to anticipate potential toxicity problems. So we discussed modes of actions, creating artificial neural networks, showed some examples of R models and in ADMET Predictor under the toxicity module, and then finally showed the tox risk score. And so thank you, and I'll turn it back over to John.
Thank you, Michael, for a very enlightening presentation. We'll now begin the question and answer portion of today's webinar. You may either type in your question into the questions pane, or if you'd like to ask a question using your telephone, please use the hand raising icon on your control panel and be sure to enter your unique audio PIN. There are a number of questions that have been submitted during the course of the webinar, and so I'll start with the very first one. How do you define toxic versus non-toxic in the classification models? Is that an input parameter that's defined by the researcher? We also have, I'm sorry, Michael, before you answer, also Bob Clark, our Director of Life Sciences, is now on the call and will be available to help answer questions.
Okay, that's an excellent question. Sometimes it's defined by the researcher. Other times we have to predict a cutoff. So we look at the distribution of the values and see if there's a native or when we look at the distribution, if it's a bimodal distribution, maybe that tells us to have the cutoff between the two peaks in the distribution. So, for example, in the HERG model for classifying into either as toxic or non-toxic, I think we picked the value of 1 micromolar or less to indicate toxicity. So it is a difficult thing or a difficult question to answer is exactly where we put the cutoff. Maybe Bob has something to add on that.
Well, for the what Michael says is very true for the actual toxicity models. In some cases, there's general agreement in the industry. For example, for mutagenicity in the Ames test, there are regulatory criteria that specify what positive means in the Ames test. In other cases, as he said, we have to. It's fuzzier, and we have to make that choice ourselves. The ADMET risk rules, many of which flag specific toxicity risks based on toxicity models. What we've done is calibrate our models against a subset of the WDI, as is shown in the slide that Michael's showing now, such that 90% of the compounds do not trigger the toxicity flag. I hope that answers the question.
Thank you, Michael. Thank you, Bob. Next question, and I believe this was answered during the course of the presentation, but just to clarify, are there any in silico validated models for predicting endocrine disruptors?
Yeah, the androgen receptor and estrogen receptor models.
They are currently available in the ADMET Predictor software, yes?
Yes.
Okay. Another question is related to predicting renal toxicity. Is there any thought about moving forward with predicting renal toxicity in ADMET Predictor?
We do not currently have a renal toxicity model in ADMET Predictor. Bob, can you address that question better, or maybe you, John?
I would say that I'd encourage that user, I don't remember which one it was, and anybody else in the audience that thinks that would be a useful model, to send us some details about what kind of toxicity you mean. Do you mean a very, very generalized model? akin to the hepatotoxicity model, or a specific, for example, a specific transporter blockade. So we'd be happy to take input on what we should do in that regard, but we don't have anything right now, and we do not have anything in preparation at this point.
Thank you, Bob. A question that came up while you were demonstrating ADMET Predictor, Michael, is how are new chemical species actually entered into the system for prediction? What kind of file formats, I think, are people able to load into the software?
SMILES files, SMILES strings, or SD files, SD formatted files can be read in ADMET Predictor.
Are neural networks the only type of learning algorithm that is available in ADMET Predictor?
No, we have several methods. So in the Advanced Modeler tab, so I was in artificial neural network models, but you can also create support vector machine models, KPLS, and then finally multiple linear regression models. So all those algorithms are available within ADMET Predictor.
And I would emphasize that all the models, by default, you can turn this off, but all of them are actually ensemble models. So you actually generate multiple separate models, and then the prediction is an average of some kind of those individual model predictions. None of the models we distribute to users are single artificial neural nets or single SVMs.
Thank you, Bob. Thank you, Michael. Can you use these models to determine the commonalities of one set of chemicals compared to another set of chemicals? I believe this is probably related to similarity, maybe loading in one set of chemicals and seeing how these compounds compare to perhaps the chemicals used to build models, for example. I don't know if you want to focus a little bit on similarity features in MedChem Studio, or discuss a little bit about, again, the process of determining applicability domains.
I guess there's different ways to do it. One way might be to compare the molecules using a Cohonen map. So a Cohonen map is based on the descriptors of the molecules. So you have descriptors for each sets of molecules, and you can see if they fall into similar cells in the Cohonen map. Another way is to use our 4D data modeling. So with this, you can create a principal. You can perform principal component analysis of the molecules using our descriptors, and so you get you could graph the components and say color one set of compounds one color, or points of one set one color, and points of another set a different color, and that'll allow you to visualize if there's overlap between the two sets of data.
Michael, can you click on basic modeler settings? I want to also comment on similarities. In the right panel, you can see the list of the descriptors. So using that list, you can determine similarity in descriptor space using Cohonen map. But there's nothing preventing you to determine similarity in terms of properties. So you can select all the descriptors, move it to the left panel, and select all the models from the left panel, moving to the right. Okay? And that will make all the model predictions, or a subset of predictions, available for similarity testing.
And you can also visualize the similarity using the 4D data mining.
Yes, exactly.
tool.
You can do the same with 4D data mining tool. And in addition, our tool MedChem Studio has a very rich range of similarity methods, clustering methods, classification, and so on. So the possibilities are virtually endless.
Thank you, Bob, Robert, and Michael. Another question is, are the PD50 carcinogenicity models sex-specific?
No, they are not.
No, they're not.
I will say we have a collaboration going with the CFSAM division of FDA, where we have been looking at doing that. Several people have, and the problem is that the data are not real reproducible, and so it's a real challenge. We have looked at doing that.
Thank you, Bob. Another question, how do you interpret complex toxicity endpoint developmental carcinogenicity predictions when using such a large number of descriptor variables?
Could you repeat that question, John?
Sure. How do you interpret complex toxicity endpoint developmental carcinogenicity predictions when using such a large number of descriptor variables?
Oh, how do we interpret it? Go ahead.
You can show the descriptor sensitivity analysis tool, I think.
Yeah, okay, so we have a tool called Descriptor Sensitivity Analysis, and so you can select a particular model. So I think it was some kind of carcinogenicity. Was it development or... so I'll just pick any model here. So here it's rat toxicity. Robert, go ahead.
Yeah, go ahead. You're doing fine. This
Okay,
illustrates well that in spite of the large number of descriptors, only a handful are significant for per compound. That's important distinction, right? That it's per compound because the model is non-linear. The sensitivity must be determined in a specific point in space. Okay, so for this first compound, we have, I would say first 10 descriptors being significant, and the rest of the descriptors are diminishing in terms of sensitivity. So interpretation is really easy in spite of the appearances. So you can, let's say, see that, for example, number of phosphorus atoms is highly significant for this compound, and you can click on the sensitivity line and see how that property depends on a given descriptor. It's still updating. So, okay. So here it is. Okay, it's just single descriptor curve. Okay. That means how the model is sensitive with respect to the descriptor, keeping all other descriptors constant. Hope that answered the question.
Thank you, Robert. Thank you, Michael. Another question is, do you have any plans to work with OECD to fulfill REACH requirements?
Bob or Robert, do you want to answer that one?
Okay, so essentially our models fulfill all the REACH requirements except one. Okay, that is make the models completely transparent. That means revealing everything about the model, which I think is a common denominator to all commercial vendors who supply such predictive models. Okay. No, it really is. This requirement is at odds with the desire of the vendor to keep the intellectual property protected from competitors. Okay. And I personally don't think that this requirement is very critical, because if model is well defined and if it's available to anyone who wants to evaluate it, okay, I don't see any problem with having some of its details proprietary. I really hope that OECD will relax this requirement. Otherwise, the whole segment predictive modeling community is being shut off.
Okay, thank you, Robert. Another question, with respect to the mutagenicity models, there are a total of 10 models. How come the score given in the tox mute risk column is only between 0 and 6? If there are 10 models, one would expect the max score to be 10. Can you please explain?
Yeah, it's rare for a compound to be toxic in all 10 of the models, both experimentally and our predictions. So here's the 10 scores on my screen. So there's not many compounds that will be positive for every one of the strains.
I think the question, though, is going to be why does the mute risk score only range from 0 to 6, as opposed to each individual model getting a score of 1, making the maximum equal to 10, potentially?
Oh, okay.
It does not. I don't know where this number came from. Michael, you can actually go to Tools, Model Editor. You can bring up the tox score, and it should range from 0 to 11, not 6.
No. Tox. Wait. Tox risk is for all toxicities. He's not talking about mute risk. He's talking about tox risk.
No.
Yes, I know. It's so click on toxicity risk.
Mute risk.
Filter. Yeah.
Mute risk.
Yeah,
and create edit ADMET risk button. Yeah, right here.
Okay.
So you can see that you have 10 rules for each tox model and 2 mixed rules for mixtures of tox models. Okay, so you look at weights, right? The 10 models have weight equal to 1, and the 2 rules that are extra have weight equal to 0.5. So in theory, the range is from 0 to 11. I know where 6 came from. This is correct.
It's because that's what the tooltip says.
Yes.
And I think it's because there's redundancy.
Oh, the tooltip is wrong. I'm sorry.
Well, there are 5 strains, right? And there's redundancy, so that if you get a positive prediction without S9 being added, then you expect to get it be positive with S9.
That's true. The models.
I believe that's why.
Okay. I understand that. The models are trained independently, though.
But the rule takes that into consideration. Is that you don't double count a strain if it's in with and without S9, because that just says that strain is sensitive.
Oh, you're right.
It distinguishes 2 different strains.
And not. Yeah, there is a qualifier. Yeah.
So it's a little more subtle.
That's right. That's right. Because you're right. There are these. So Bob is the author of the Tox mute risk.
So the reason it's 6 is that there are 5 strains, even though there are 10
models.
Models.
Yep. There's all the additional qualifiers and not.
And in fact, there are several of the ADMET risk rules where that sort of interdependence between the outcomes is factored into how the rules are formulated.
Thank you, Robert. Thank you, Bob. Just a couple of more questions here. One is, and I'm not sure if this has already been covered or not, but can we determine the toxicity for specific chemicals and their metabolites in particular organs?
In heart, for example, we have a very model, but a liver toxicity. So not all the organs, unfortunately. We cover some organs, heart and liver. I don't see we have any other specific models,
reproductive
endocrine
toxicity.
which is reproductive toxicity. It's general. So the answer is partially yes.
Okay, heart, liver would be organs in which we have models already.
Yes.
Okay.
Oh, actually, there is one caveat to that. Actually, if you know your clinical chemistry, the individual hepatotoxicity models, there are some combinations of those 5 or 6, I forget, the blood tests, which they don't set a hepatotoxicity risk flag, and that's because they're actually indicative of other problems. Lactate dehydrogenase, if that's predicted to be elevated, and none of the other liver enzymes are predicted to be elevated, that's a suggestion that you are likely to get muscle damage. Just the way the blood test would be.
Yeah, and you're right. And Michael, you can actually open now, close that box risk and open a SIP risk. And let the audience take a look. Model editor.
Oh, I'm sorry.
Model editor SIP risk, where rules are created in exactly the way that
I have it up.
I'm sorry. I'm dreaming here. Tox risk. Close that. Close that. I'm sorry. Tox risk. It's there you go. Tox risk. Yes. So exactly. In terms of liver toxicity, we have complex set of rules labeled HP and SG. Which take these enzyme interactions into account.
Yeah, but the other combinations are ones that are not as critical, so we haven't built them into separate risks. There's a very rare bone condition that I think elevates alkaline phosphatase and lactate dehydrogenase, and nothing else.
Right.
Thank you, Robert. Thank you, Bob. There are a couple of other questions that are outside the scope of today's webinar, and we're already at 1 hour and 15 minutes. So we will be following up individually with the people who asked specific questions about the simulation module and also about the possibility to do a webinar which combines the use of ADMET Predictor with Gastric Plus to do some in vitro and vivo extrapolation work. Right now, just to close the web meeting, what I'd like to do is just send it quickly back to Anu, who will just give some brief information about where you can see us next.
Hello, I'm back. So I would just briefly introduce you to our 2 weeks of free evaluation licensing offer. So you can contact me for ADMET Predictor and MedChem Studio. My contact details are in the slide, and we will be following up with the slides as well, and you can contact us on our website as well if you would like to evaluate. ADMET Predictor, you can reach me for Gastric Plus and for Ddv Plus. Please contact John DeBella. And if you have a small project that you would like to get a consultancy services, Michael Lawless will be your point of contact. Next, if you are in the Bay Area, San Francisco Bay Area, Foster City, we will be exhibiting, and also we will be the major sponsor at the upcoming PACO PBS meeting on December 10th, which is Monday. And please stop by our booth, and Michael Bolger will be giving a talk on other applications of simulations plus products. And we are also holding a series of workshops in the first half of next year This is our first workshop is going to be on the ADMET Design Suite and the Gastric Plus in Boston. So if you're on the East Coast, this is a great opportunity for you to connect with us and learn more in depth about the course. So this is about a week-long course back-to-back on ADMET Predictor, Design Suite, and Gastric Plus. And also for our West Coast clients, we are holding a Gastric Plus workshop in San Diego in April from the 22nd to the 24th. So if you're on the West Coast, please contact us on our website and register for these workshops. With that, we really appreciate your time and your attention. Like I said, I will be following up in the emails, and if you have any questions, please don't hesitate to contact me or to reach our website and contact John with any further details. Over to you, John.
Thank you, Anu. And thank you again, Michael, for a very enlightening presentation, and Bob and Robert for your comments. That's going to conclude today's webinar. If you did miss any part of today's presentation, the playback will be available at our website, www.simulations-plus.com. Thank you so much for joining us today, and have a wonderful day. Take care.